All right, so today we're taking a deep dive into something pretty fascinating, advanced analytical techniques in drug development and quality control.
Yeah, it's all about the science behind making sure the medicines we all rely on are safe and effective.
Yeah, and that each batch is consistent.
Exactly. Consistent quality.
And to really dig deep into this, we've got a whole mountain of sources.
We've got everything from pharmacokinetics, you know how drugs move through the body?
Right, how they get absorbed and broken down and all that.
Yeah, exactly. To drug interactions, how different meds can affect each other.
Which can be a really big deal clinically.
Absolutely.
And then there's the whole world of high-throughput screening, where they're testing tons of compounds really fast to find new drug candidates.
Yeah, that's where automation and robotics come into play.
And we'll also look at how artificial intelligence is starting to change the game in drug development.
AI is really making waves in so many areas.
And then, of course, we can't forget about the crucial steps of preclinical and clinical trials.
Right. Making sure new drugs are safe and effective in humans.
We've also got sources on the nitty-gritty of medicinal chemistry.
Yeah, how they actually design and synthesize the drug molecules.
And then even things like how lipids crystallize and how tablets get their coatings.
Sounds pretty specialized, but those details can really matter for how a drug works.
Absolutely. Plus, we're looking at analytical science in general, soft gel capsules, new formulations, bioequivalence studies, nanoparticles for drug delivery.
Wow, we've got it all.
And of course, all the regulations and guidelines like the Code of Federal Regulations, ICH guidelines, and articles from the FDA.
So basically, everything you need to know about how they make sure drugs are up to snuff.
Exactly.
Our goal in this deep dive is to break all this down and make it clear and engaging and show you just how much these analytical methods matter.
Right. We want to reveal the hidden power of these techniques.
And offer some valuable insights along the way without getting too bogged down in technical jargon.
So let's jump right in, shall we?
Let's do it.
When we talk about these advanced analytical techniques, where do we even begin?
Well, at the very heart of it, you have a few core technologies,
Okay, like what?
like chromatography, spectroscopy, and mass spectrometry.
Chromatography, that sounds kind of intimidating.
It might sound complicated, but think of it like this. It's a super refined way to separate mixtures.
Okay, so like if you had a salad with lots of different ingredients.
Exactly. Chromatography would be like separating out all the lettuce, tomatoes, cucumbers, and dressing so you could examine each one individually.
Ah, so in pharmaceuticals, they're using it to isolate the actual drug from everything else.
Right, whether it's from a formulation or a biological sample like blood.
That makes sense.
Chromatography is all about getting that pure compound so we can analyze it.
Okay, and then what about spectroscopy? What's that all about?
Spectroscopy is all about how light interacts with matter.
Light.
Well,
Interesting.
Yeah, so by analyzing the spectrum of light that a sample absorbs or emits,
Absorbs or emits.
we can get a fingerprint of what it's made of and its structure.
So it's like each molecule has its own unique way of interacting with light.
Precisely. And we can use that to identify and quantify it.
Pretty cool.
One very common type of spectroscopy is UVV's spectroscopy, which uses ultraviolet and visible light.
UVVs, I've heard of that, but I'm not really sure what it's used for.
Well, it's often used to figure out the concentration of a drug in a solution, for example.
Oh, so like how much of the active ingredient is actually in that pill or injection?
Exactly. It's all about precise measurement.
And you know, I actually saw an example of this in one of our sources.
Oh, really? Where was that?
In pharmaceutical analysis, there's calculation example 4.3.
Ah, yes. That's a good one.
It shows you step by step how to calculate the concentration of a drug using UVV spectroscopy.
Yeah, they take you through how much the sample was diluted and its absorbance reading on the spectrophotometer.
Right, and from that, you can actually calculate the exact amount of the drug in the original sample.
It's a very practical illustration of how this technique is used in real-world pharmaceutical analysis.
Very cool. And then there's mass spectrometry, which always sounds so high-tech to me.
It does have a certain ring to it, doesn't it?
Yeah, like something out of a sci-fi movie.
But it's a very real and powerful technique.
So what exactly does mass spectrometry do?
It identifies and quantifies molecules based on their mass-to-charge ratio.
Their mass-to-charge ratio. Okay, break that down for me.
Basically, it's like putting individual molecules on a super sensitive scale.
So we can see how heavy they are.
Exactly. And by knowing their mass and charge, we can figure out exactly what molecule it
It's
is.
like a molecular fingerprint.
You got it. And not only can we identify what's in a sample, but we can also determine how much of each molecule is there.
That's amazing. So we're not just getting a qualitative analysis, we're getting a quantitative one too.
Precisely. And often mass spectrometry is used in combination with chromatography in techniques like LCMS or GCMS.
LCMS, GCMS, those sound familiar, but remind me what they stand for.
LCMS is liquid chromatography mass spectrometry, and GCMS is gas chromatography mass spectrometry.
Ah, okay, so we're combining the separating power of chromatography with the identification and quantification power of mass spec.
Exactly. It's a 1-2 punch for analyzing complex mixtures.
So we're getting a really detailed picture of what's in those samples.
Absolutely. These techniques have revolutionized how we study drugs.
Okay, so we've got these core techniques, but how have they evolved to keep up with the demands of modern drug development?
They're constantly being refined and improved, becoming more sensitive, more accurate, and faster.
Because drug development is getting more complex all the time, right?
Absolutely. We're dealing with increasingly complex drug molecules and biological systems.
So we need cutting-edge analytical techniques to keep up.
For example, in pharmacokinetics, where we're studying how a drug moves through the body,
Yeah, like how it gets absorbed, distributed, metabolized, and excreted.
we need super accurate measurements of drug concentrations in blood plasma over time.
And those measurements are crucial for understanding the drug's journey through the body.
Precisely. It's like plotting the drug's course on a map. And if those measurements are off, the whole map is skewed.
I see. So data accuracy is absolutely paramount here.
It's the foundation upon which everything else is built.
And it's not just about pharmacokinetics, right?
No, these advanced techniques are also critical for in vitro studies.
In vitro, meaning in the lab, outside of a living organism.
Right, so we might use in vitro studies to predict how different drugs might interact with each other.
Because sometimes taking multiple medications can cause unexpected problems.
Yes, that's right. And by carefully analyzing how different drugs are metabolized by enzymes in the body, we can anticipate potential interactions.
So it's like a preemptive strike against drug interactions.
Exactly, we're trying to catch those potential problems before they ever reach patients.
That's got to be a huge help in ensuring patient safety.
Absolutely. And these techniques are also essential for high-throughput screening.
High-throughput screening, that's where they test thousands, maybe even millions of compounds,
It's like a massive fishing expedition for new drug candidates.
casting a wide net to see what they can catch.
And without these advanced analytical techniques, it would be impossible to screen that many compounds efficiently.
Right. They need to be able to analyze samples and generate data really fast.
Speed and accuracy are key in high-throughput screening.
It's mind-boggling to think about the scale of the analysis they're doing.
It is pretty incredible how far we've come.
And now they're even combining all this analytical data with other types of biological data.
Yeah, it's called multiomics, or integrative analysis.
So instead of just looking at the drug itself, we're looking at how it interacts with the whole biological system.
Exactly. We're integrating information from the genome, epigenome, transcriptome, all alongside the analytical data on drug activity and metabolism.
So it's like taking a holistic view of the whole picture.
Precisely. And this can reveal subtle disease mechanisms that we might have missed before.
It's like moving from a single snapshot to a full-length movie.
And that can lead to more targeted and effective therapies.
Now, I want to shift gears a bit and talk about regulations, because I know the FDA has some pretty strict standards for drug development.
Oh, absolutely. There are tons of regulations and guidelines that companies have to follow.
And I'm guessing these advanced analytical techniques play a big role in ensuring compliance.
They're absolutely critical. They provide the objective data needed to demonstrate that drugs meet all the necessary quality standards.
So it's not just about making a drug that works, it's about proving that it meets all the safety and quality requirements.
Exactly, and that documentation is crucial.
So what are some of the specific regulations where these techniques come into play?
Well, for example, 21 CFR Part 111 emphasizes the importance of establishing precise specifications throughout the manufacturing process.
Specifications, meaning like a set of criteria that the drug product has to meet.
Right, like purity, potency, stability, all those things.
And these specifications have to be in place at multiple points along the way, not just for the final product.
Exactly. It's about ensuring quality control at every stage.
And to prove that those specifications are being met, companies need to validate their analytical methods.
Validation is a rigorous process to demonstrate that a test is accurate, sensitive, specific, and reproducible.
So basically, they're proving that their tests are up to the task.
Right. They need to provide solid evidence that their analytical methods are reliable and fit for purpose.
And even if they're using well-established tests from official sources,
They still need to verify that those tests work correctly under their specific conditions.
so it's not a one-size-fits-all approach. They have to tailor the testing to their own manufacturing process,
Exactly. And all of this applies to all types of pharmaceutical products.
including those highly specialized drugs like PEEP drugs, right?
Two drugs, you mean those used in positron emission tomography?
Yeah, those ones, because they have a really short shelf life.
Very short half-lives, yes.
So I imagine rapid and accurate analysis is even more critical in those cases.
Absolutely. There's no room for delay when you're dealing with radioactive materials.
So analytical techniques are not just about ensuring the quality of the final product, they're also essential for keeping patients safe during the manufacturing process.
Right. They provide the data needed to make informed decisions about when a batch is ready for release.
Now, let's talk about how these techniques contribute to ensuring the quality of the final drug product.
Well, even in the very early stages, like preclinical screening of potential new drugs, analytical methods are used to understand how the drug might behave in living systems.
Preclinical meaning before they start testing in humans.
Correct. And in preclinical studies, they're looking for things like how the drug is absorbed, distributed, metabolized, and eliminated.
And they're also trying to predict potential mechanisms of drug resistance.
Especially in anti-cancer drug development, drug resistance is a huge challenge.
So they're using analytical techniques to identify potential roadblocks early on
Exactly. They want to anticipate and overcome these challenges as early as possible.
to make sure the drug is going to be effective in the real world.
Precisely. And then when they move into bioequivalence studies,
Bioequivalence studies, that's where they compare a generic drug to the brand name drug
right, they want to make sure the generic version delivers the same amount of drug to the body at the same rate.
to ensure that it's truly interchangeable with the original.
Exactly. And this is where highly accurate analytical measurements are crucial.
They need to track the drug's concentration in the blood over time to see how the two formulations compare.
And those comparisons are based on the data generated by these sophisticated analytical techniques.
So it's not just about making a drug that works. It's also about making sure it's formulated in a way that delivers the drug effectively.
And that's where formulation development comes in, which is all about designing the best way to present the drug to the patient.
So we're talking about things like tablets, capsules, injections, creams, all those different forms.
Exactly. And analytical techniques like freeze-drying microscopy and subambient dynamic mechanical analysis are crucial for formulation development.
Those sound pretty specialized.
They are, but they help determine critical temperatures and other physical properties of the drug substance in the formulation.
Which can affect things like stability and shelf life.
Absolutely. And those factors are crucial for ensuring the drug remains effective until it's used.
And I imagine analytical techniques also come into play in monitoring for impurities during the manufacturing process.
Oh, absolutely. Impurities can arise from various sources during manufacturing.
Like from the raw materials, the equipment, or even the manufacturing process itself.
Right. And as they scale up production from small batches to large industrial quantities, the types and levels of impurities can change.
So they need to be constantly vigilant and monitoring for these impurities.
And that's where analytical techniques like LCMS and GCMS are essential.
They allow them to identify and quantify these unwanted substances, even in trace amounts.
And based on that data, they can develop purification procedures to remove those impurities,
So it's a continuous cycle of analysis, purification, and refinement.
all driven by the power of these analytical tools.
It's amazing how much these techniques impact every aspect of drug development and quality control.
And it doesn't stop there. Analytical data is also crucial for controlling the physical properties of the drug substance itself.
Physical properties, you mean things like its crystal structure and particle size.
Exactly, because even small differences in those properties can affect how the drug dissolves and gets absorbed in the body.
So they need to make sure those properties are consistent from batch to batch.
Precisely. And they rely on analytical techniques like differential scanning calorimetry and particle size analysis to ensure that consistency.
It's clear that analytical science is the backbone of the entire pharmaceutical industry.
It really is. It underpins everything from drug discovery to quality control to regulatory compliance.
Now, to bring this all to life, can you share some real-world examples of how these techniques are being used?
Of course, in the search for new antibacterial agents, for example, they use analytical techniques to determine the minimum inhibitory concentration, or MIC, of compounds.
MIC, That tells you how potent an antibacterial compound is against specific bacteria.
Right, it's a key measure of its effectiveness.
So they're using analytical techniques to evaluate the potential of new drugs.
Exactly. And in pharmacovigilance, which is all about monitoring drug safety after they're on the market.
Right, because sometimes problems don't show up until a drug is being used by a large population.
Exactly. And in those cases, analytical methods can help identify the cause of any adverse drug events.
So they might analyze patient samples to look for unexpected metabolites or degradation products.
Right, they're using those analytical tools to investigate and understand any safety issues that arise.
It's like a detective work trying to piece together the clues.
And in the study of drug metabolism, analytical techniques are essential for characterizing how drugs are broken down in the body.
Because understanding drug metabolism can help predict potential toxicities and drug interactions.
Precisely. And advanced mass spectrometry is a powerful tool for unraveling those metabolic pathways.
It allows them to identify any potentially harmful byproducts that might be formed.
Right, they're mapping out the drug's journey through the body and looking for any potential hazards along the way.
And then there's the issue of removing trace amounts of catalysts used in drug synthesis.
Ah, yes. Catalysts are often used to speed up chemical reactions, but you don't want any residual catalyst left in the final drug product.
Right, especially if it's a heavy metal or something like that.
So highly sensitive analytical techniques are used to monitor the levels of those residual metals.
To make sure they're below the acceptable limits.
Absolutely. Patient safety is always the top priority.
And those analytical techniques are also used to assess the stability of drug products over time.
Right. They need to make sure the drug remains stable and potent during storage and transportation.
Because you don't want it degrading before it reaches the patient.
Exactly. And they use those analytical data to determine appropriate storage conditions and expiration dates.
And finally, they're using analytical methods to analyze biomarkers. Biomarkers are molecules that can indicate disease state or predict a patient's response to a drug.
And by accurately measuring these biomarkers, they can improve patient selection in clinical trials and move towards more personalized medicine.
So instead of a one-size-fits-all approach, we're getting closer to tailoring treatments to individual patients.
It's a really exciting area of research.
It's incredible to think about how much these analytical techniques are doing behind the scenes to ensure the quality and safety of our medicines.
They're the unsung heroes of the pharmaceutical world.
They're essential for generating accurate data, ensuring regulatory compliance, and ultimately protecting patient health.
They're the foundation upon which all of modern drug development is built.
So as we wrap up this deep dive, it's clear that advanced analytical techniques are much more than just lab instruments.
They're indispensable tools that touch every aspect of the drug development life cycle.
From the earliest stages of discovery to the final product that reaches patients.
These techniques ensure that the medicines we rely on are safe, effective, and consistently high quality.
And they're constantly evolving, becoming more sophisticated and powerful all the time.
Which leads us to a thought-provoking question for our listeners.
As these analytical technologies continue to advance, particularly with the integration of artificial intelligence,
How might these advancements further revolutionize our understanding of drugs and accelerate the development of new therapies?
and what new challenges might we face in ensuring the reliability and interpretation of these increasingly complex data sets?
It's an exciting time to be in this field, with so much potential for innovation and discovery.
But it's also important to be mindful of the ethical and practical implications of these advancements.
We need to make sure we're using these powerful tools responsibly and for the benefit of all.
It's a fascinating area to ponder, and we encourage you to explore it further.
Thanks for joining us on this deep dive.
We hope you found it informative and engaging.